Okay, so can you hear me clearly right now, since this is a live video? You can hear me, right? Okay. So thank you very much for the very nice introduction and the kind invitation. It's really a pity that I cannot be there in Italy, although I have lots of person I hope to discuss with in person, but let's save it for the future. So like in the introduction, so today I will discuss the sperm RNA code in epigenetic inheritance, which at its core, it's about the sperm RNA population and their modifications and how they might be able to transfer the phenotype to the next generation. So now when we talk about the phenomenon of epigenetic inheritance or the inheritance of acquired traits, so more times than ever, we were actually discussing about another very fundamental topic, which is about what is a hereditary information carrier. And perhaps more specifically, we were discussing, are there anything that beyond the DNA sequence that can act as an information carrier? And the reason is because the DNA itself, it's pretty much like a read-only disk. So we can get information from it, but cannot easily write information in upon certain environmental fluctuations. For example, if we have a big meal or have an emotion, it's very unlikely that this type of environmental fluctuation will change the DNA sequence, especially change the DNA sequence in the germ cell or perhaps in the sperm. So this is to say, if we only consider DNA sequence as the hereditary information carrier, this will almost rule out the possibility of the inheritance of acquired traits from the environment. But now with the emerging topic of epigenetics, now we understand that in addition to the DNA sequence, which is still the backbone in this modern version of Lamarck's giraffe, which was designed by the very talented members in our lab. So now you can see that in addition to this backbone, there are other gadgets in this DNA. For example, this DNA methylation, and these are histone marks, and the different color represent different modifications. And also there are RNAs, RNA molecules, which is also modified, and they can act in trans to interact with this backbone of the DNA and these marks. So this is to say, when we consider this picture with these gadgets, it becomes much dynamic, because many of these marks, like the DNA methylation, they have enzymes to add it and has enzymes to erase it, and also proteins to read it. So we can consider if there is certain environmental exposure that can change this enzyme that can act on these marks. So these marks might be changed upon this environmental fluctuation. So this just enables the possibility of this Lamarck's giraffe and the inheritance of acquired traits. So basically, this is the idea, and I hope following this idea, I will introduce a little bit more on the actual evidence. So this was a classic experiment that actually drive me into this fascinating field. So in this model, using either a rat or a mouse, so if we feed the rat or mouse under a high-fat diet over a certain period of time, so apparently they will become obese and develop metabolic disorders. So these are not news, but the intriguing part is that if we let this male mice mate with a female mice or rat that are fed under a normal diet, then their offspring will automatically develop metabolic disorders, even if they were fed on a normal diet. So this experiment strongly suggests that the sperm of the father during this environmental exposure were absorbing some information from the environment and relate this paternally acquired phenotype to the offspring, which is metabolic disorders. So this is fascinating. But after the series of experiments were first published, there are still skepticisms floating around. For example, some scientists will argue that perhaps it is because of the mating process, the male is transferring something else to the female. For example, what if the microbiome of the female were changed, or perhaps the eating habits of the female were changed, and that leads to the phenotypical outcome of the offspring. Basically, people will argue that the resulting offspring phenotype might be result from the female part but not the sperm. So to eliminate these confounding factors, later on, more stringent experiments were conducted, including using in vitro fertilization. And in our case, we have isolated the sperm to perform the ICSI. So this has completely eliminated the male-female interaction. So as a result, the resulting offspring still can have a metabolic disorder during the growth. So in my opinion, this series of experiments has strongly demonstrated that the sperm by itself can really absorb some information during this paternal environmental exposure. And in this case, it's the heifer diet. So the real question becomes, what has been changed in the sperm? As we discussed before, most likely it's not the DNA sequence that has been directly changed. So most probably it is the epigenetic information carriers that has been changed in the sperm. Now let's take a look into the sperm and what are the potential epigenetic information carriers. As we mentioned before, there could be DNA methylations and histone marks and the 3D structures and also the RNAs and RNA modifications. Now, right now, I know that different labs have their own favorite in regards to these different molecules. But before we considering any of these single molecule as the causal factor in this epigenetic inheritance, let's remember one very fundamental fact, which is called epigenetic reprogramming, which will happen right after the fertilization of the sperm and the fusion of the sperm and the egg. And during this process, most of the DNA methylation and histone modifications will be first wiped out, followed by reestablishment. So this fact actually raised another layer of skepticism in the field, that how could certain phenotype could be transmitted to the next generation if all these marks has been removed. So of course, there are arguments that some of the marks are more resilient than others. For example, the imprinting loci will be protected from being erased. And also some other marks will be first erased and then reestablished. And in our lab, we are more interested in studying the sperm RNAs, and for good reasons. One reason is because that the RNAs, they are mobile. They will not be immediately erased. And in fact, the RNAs can act in trance, can interact with DNA and histone marks. So they might be able to facilitate the escape or perhaps the reestablishment of some of the marks. So these are the hypotheses. And another more direct reason is because the sperm RNAs, the RNA population in the sperm are very unique, and especially the small RNA populations. So this was a paper that we published about 10 years ago. So this was an exploration trying to identify the small RNA populations in the mature sperm. So after we sequencing the sperm, the mature sperm sample, which is from mouse, we realized that in addition to the very popular micron species, we have found a new peak. And this new peak, they are not pi RNAs. After some in-depth analysis, we have realized that many of these sequences are coming from mature tRNA. So we have chosen to call them tRNA-derived small RNAs, or tsRNAs in short. Actually, there are other labs simply call them tRNA fragments, but basically we were talking about the same molecule. And in that first paper, actually, we took a very hard time to get it published because this is very new. By the time we want to publish the paper, the reviewers were asking us a question that whether these RNAs can be really delivered at the time of fertilization. So actually, this was a question that cannot be easily addressed, but we have chosen to address it in an indirect way. So we have chosen to purify the sperm heads and demembering it, so only leave the nude nucleus. So we use the nude nucleus to extract the RNA and perform the PCR analysis of these tsRNAs. And we have again detected lots of tsRNAs in the head, which suggests that these tsRNAs are deeply embedded in the sperm nucleus. So by using this indirect evidence, we have convinced the reviewer that these RNAs, because they are deeply embedded in the head, they must be delivered at the time of fertilization. So We get this published about 10 years ago. And in that same paper, we have also profiled the dynamic changes of the sperm RNA during the spermatogenesis and epididymal transition. So as summarized here, we have found that there is a very dynamic picture during this spermatogenesis and epididymal transition. During the spermatogenesis process, which is inside the testis, the majority of the sperm RNAs are pi RNAs, but in the epididymal sperm, the majority of them are TS RNAs. So that was a conclusion we reached more than 10 years ago, and we were happy to draw this figure a few years ago. But right now, I can tell you that this figure is incomplete. And the reason is that later on, we have realized that many of the tsRNA populations, they are highly modified because they contain different types of modifications. And some of these modifications will prevent the library construction during the normal sequencing process. So in other words, these highly modified tsRNAs, they will escape the library construction. So they will not be detected during the sequencing result. It's like they are wearing an invisibility cloak. So to conquer this problem, we have published a paper. This was last year. We developed a new method, which is called Pandora-seq. This method will use a series of enzymatic treatments to first eliminate these modifications, and after which we can detect more tsRNAs. Now we can see from this figure that comparing to the microRNAs, the tsRNA population are even more. But the most surprising part in this paper was that in addition to the microRNAs and the tsRNAs, we also detected lots of RNAs. And what is RNAs? These are RNA-derived small RNAs, which means they derived from ribosomal RNAs. So actually, I can tell you that at first we were not very happy about this figure because comparing to the RNA population, both microRNAs and tsRNAs become a minority. So where is my favorite tsRNA? But this is the reality we have to accept. So actually, after a series of Northern blot validation, we have found that indeed the RNA is the most abundant small RNA populations in the sperm small RNA populations. So our knowledge continuously evolving. So also in this very paper, we have chosen to do the trick one more time. So we have chosen to isolate the sperm head and demembranize it and only leave the nucleus. And we have performed Pandora-seq again. But now you can see that when we detect the sperm RNAs that are deeply embedded in the head, now the tsRNA population become much more abundant. So this is very intriguing. So which means that the sperm small RNA population in the are highly compartmentalized. And this is physiologically relevant because we can imagine that the sperm RNA in the cytoplasmic compartment, their quantity are relatively small, especially when comparing to the size of the oocyte. The RNA quantity in the sperm is almost infinitesimal. But when we consider the RNAs that are deeply embedded in the nucleus is relatively compartmentalized, even after the fertilization process. So it is, in our opinion, so perhaps these sperm RNAs deeply embedded in the head can engage more during this epigenetic reprogramming. And actually, this is a new direction that our lab is actively exploring. So I know the audience might not only work on sperm, so I just want to mention that using Pandora-seq, we have performed lots of tissue samples and cell samples. We have found that, like here, in traditional RNA-seq, the majority of the sperm RNA remains to be microRNAs. But if you perform Pandora-seq, we can detect many more tsRNAs and RNAs. And this is the example of brain. And now we can take another example of the embryonic stem cell. We can see that in Pandora-seq, the microRNAs is actually a very small minority. And the majority of small RNAs are actually tsRNAs and RNAs. And in this very paper, we have some proof of principle study demonstrating that both tsRNAs and RNAs can regulate the cell lineage differentiation in the embryonic stem cells. So I just believe that this field is just at the beginning, and using Pandora-seq, now we can detect much more abundant tsRNAs and rsRNAs, and they must have a function that awaits us to discover. So basically, in a more recent review paper, now I do not dare to draw any compositional figure about the sperm RNA or perhaps any other tissue samples, because most probably we will be wrong again, because now I believe that our knowledge is really increasing gradually. So instead of drawing a picture, we wrote a text box, which is called The Blind Man and the Elephant. This is what we truly believe. And now I truly believe, like the late President JFK once said, the greater our knowledge increases, the greater our ignorance unfolds. And I'm very happy to see that this review was highlighted as the cover story of the current issue of this journal. And just reminding us, the universe of small RNAs is so big. And previously, we perhaps were simply focusing on one galaxy like microRNAs, but there are so many more galaxies out there. So in this regards, we should develop a reference for knowledge. And in this context, the word of knowledge should be interpreted as no edge. So this is my true feeling during this exploration of the sperm RNAs. So I know this might sound a little bit doubting because we still do not understand the full nature of the sperm RNAs. But let's don't be, because even without the full understanding of the composition of sperm RNAs, we can still do very important things. For example, there was a very critical question in this field of epigenetic inheritance, which is whether the sperm RNAs alone can transmit the phenotype of this acquired phenotype from the paternal exposure. So right now I can tell you that the answer is yes, at least for the dietary changes and the mental stress exposures. And here I will show you the original settings of these experiments. So actually there were 3 independent labs across continents have done very similar things. And we have been using very different models. And one lab has been using this mental stress. And in our lab, we have been using paternal high-fat diet. And in another lab, they were using high-fat, high-sugar diet. So in all these experimental settings, we have chosen to first expose the father and then extract the sperm RNA from the exposed male and inject them into a normal zygote, and followed by phenotypical tracing in the offspring. So in all these data, we have found that the offspring will either partially or fully mimic the phenotype of the father. And in our case, we have only found a partial penetrance of phenotype, which is after the sperm RNA injection, we can see the transmission of glucose intolerance, but not insulin resistance in the offspring. So this also suggests that perhaps in addition to the sperm RNA, other epigenetic marks like the DNA methylation and histone marks is also playing a role, perhaps playing a synergistic role with the RNAs. So nonetheless, it is good for us to see that the injection of sperm RNAs can have some effects in transferring the phenotype. So now the big question is, what is the effective elements in the sperm total RNA that can really do the job? So for our lab, we have a head start in addressing this question because we have chosen to dissect the sperm RNA into different fragments. For example, this 15 to 24 nucleotide RNA, it contains the majority of the microRNAs. And this 30 to 40 nucleotides, now we understand they are enriched by both tsRNAs and rsRNAs, and also these larger pieces of RNAs. So after we retrieve these different fractions of RNA and inject them one by one, we have realized that only the injection of this 30 to 40 nucleotide fraction can mimic the phenotype as that of injecting the total RNA, which means this fraction contains the essential element that can transmit the phenotype. And also, a few years later, another lab from Zurich, Switzerland, they have done a very similar result, but with small modifications. For example, they have chosen to dissect the RNA in a better resolution. And the important thing is that they have found only the injection of 30 to 34 nucleotide fraction RNA can induce the phenotype. And most intriguingly, in this phenotypical readout, they have found something very new, because in addition to the metabolic disorders, they have also found that the resulting offspring, they showed enhanced hedonic behavior, which means these mice, they love junk food and they have this alcohol preference, and because of which they have developed enhanced metabolic phenotype, which is very intriguing. Although the mechanisms we still do not understand, but this phenotype, I hope to just highlight it to show how fascinating this field could be. So I also hope to say in this very paper, in addition to injecting the RNA and performing a small RNA analysis, we have also developed a new methodology, which is called liquid chromatography tandem mass spectrometry, to analyze the RNA modification profile of these different RNA fractions. And as a result, we have found that in this 30 to 40 nucleotide fraction, 2 of the modifications shows significant changes upon high-fat diet, which is M2G and M5C. Actually, by the time we published this paper, we still do not understand what is the meaning, but we have vaguely developed this idea. These RNA modifications might be acting as an additional layer of this RNA. Perhaps they can regulate the biogenesis of dsRNAs, or perhaps they can act as an information carrier on their own. So these clues have led to the next project, which was focusing on a molecule called DMT2. I suppose that many of you might be familiar with the DMT1 or DMT3AB, because these DMTs, they can methylate DNA. But this DMT2 is quite special because the DMT2, it will not methylate DNA, but instead it will methylate tRNA at a very specific loci, which is the C38 position of quite a few tRNAs. And the intriguing part is that when this tRNA has this modification, it will become relatively stable. But when deleting the DMT2, when losing this specific M5C, the tRNA will become fragile and tends to be fragmented into tsRNAs. So this seems to be related to our tsRNA story. And because of this, we have been studying this DMT2 knockout mice under a very similar setting as we previously described, which is feed the male under a high-fat diet. But now the intriguing thing is that if we similarly inject the sperm RNAs, either the sperm total RNAs or the sperm 30 to 40 nuclear RNAs into the zygote, but this time it's from a DMT2 knockout background. And this time we can no longer induce the phenotypic outcome in offspring. So what does this tell us? This tells us that the DMT2 deletion must have done something in regards to the sperm RNA population, and making it lose the ability to transfer the phenotype in the offspring. So now the big question is, what are the changes? Now we understand that the changes at the molecular level is multifold. For example, we already understand that the deletion of DMT2 can eliminate this C38 position of the M5C very precisely. And also because of this, there is an increased level of many tsRNAs because of this loss of methylation. But in addition to that, we have also found that the deletion of DMT2 can specifically downregulate quite a few RSRNAs, and the reason for which we still do not understand. Because right now we know that DMT2 do not have a direct target on this RSRNAs or RNAs in regards to M5C deposition. So the reason that deletion of DMT2 leads to decreased RSRNAs might be something new, and it might be independent of the DMT2 methyltransferase activity. Also from this slide, I hope to mention that this non-broad exposure for the RSRNAs is only 5 seconds, and for the tsRNAs is 5 minutes or 20 minutes, but they show similar level. And again, this just demonstrated that the RSRNAs is really the most abundant species in the sperm RNA. So now, in addition to the expression of tsRNAs and rsRNAs, the deletion of DMT2 has added another confounding factors, which is because of the deletion of DMT2, the resulting tsRNAs, they can bear different modifications. And this may lead to a biological significance. For example, now we have synthesized the 3 versions of tsRNAs. One is a version without modification, another version with 5 modifications, which is mimicking the wild type situation, and another version with only 4 modifications, mimicking the DMT2 knockout situation. So from this gel, we can just tell that the different modification can alter the secondary structure of these tsRNAs, and because of which they can change their resilience to RNA degradation because of the changed secondary structure. So this proof of principle study just tell us that in addition to the sequence, the modification is another layer of control regarding the tsRNA's functionality. So basically, in summary of this DMT2 project, actually, we get a very clear phenotypical outcome, but the mechanisms is a mess. So we have coined this term, sperm RNA code, which is an umbrella term to describe the total changes of the RNA expression profile and the modification profile. So basically, we can imagine the RNA code as a 2D barcode. In this case, when the mouse was fed under a hyphen diet, it can shape a specific code that can deliver a message in the offspring. But when DMT2 is deleted, although we still have lots of information out there, but the code has been tampered, has been disrupted. So the information cannot be transferred in the next generation. So deriving from this this term of sperm RNA code, and actually this was according to the existing publications from different models, we have proposed that perhaps different arterial environmental exposure can shape a specific sperm RNA code, which can lead to a specific offspring phenotype. And now we were systematically studying more different exposures, and hopefully we'll find some common features in regards to the sperm RNA code that is responsible for delivering the phenotype. So in addition to the mouse study, I hope to emphasize that perhaps a similar sperm RNA code also exists in human beings. This was a study published a few years ago. This was a Danish study. In this study, it was shown that the lean person and the obese person have a very different sperm RNA signature. And intriguingly, if we just force this obese person to become lean by performing this gastric bypass, so shrink their stomach, so force them to lose weight. So after a few months and checking their sperm again, so they found that the sperm RNA composition is changing back in a similar fashion in that in what they found in the lean person. So which means the sperm RNA code, they are quite labile. They can be changed upon different environmental stimuli. And in a more recent case, this was a more acute treatment of in diet, which is just a 1 week exposure of high sugar diet, which can already dramatically change the sperm tsRNAs and rsRNA population. So based on these facts, so we have been proposing that we might be able to use the sperm RNA code in future precision medicine. For example, when some of the patients wants to deliver a baby, perhaps using ICSI, but right now we notice that their sperm RNA code is not in a good shape. So perhaps they will have some susceptibility in delivering metabolic disorders or perhaps even cancer susceptibility. We can give them clinical suggestions or guidance to change their diet, to enhance their exercise, perhaps over a certain period of time. So the sperm RNA code will change back to normal. Then we can recommend conception. So this is what we hope. This is a long-term dream that perhaps by doing this, we can intergenerationally prevent the disease susceptibility, which include metabolic disorders and cancers, etc. So in regards to the concept of sperm RNA code, perhaps many people, the most concerning question is that how the sperm RNA code can change the trajectory of embryo development, that leads to a phenotypical outcome. Unfortunately, for now, I think no one in this world actually understands the detailed molecular mechanisms. So our lab has done some exploratory research by injecting the sperm TSRNA enriched fraction into the zygote, and followed by transcriptome analysis, both in the early embryo and also in the offspring pancreatic islet. And we have found that in both early embryo and pancreatic islet, there were dramatic changes, especially in regards to the pathways that are relating to metabolic disorders, metabolic processes. But right now, we still do not know how this information will relate from the early embryo to the offspring. So we have some works regarding the underlying mechanisms, but now it's still not very clear. Hopefully, in the future, I can show more data. But in addition to this, overall phenomenon, I hope to emphasize one more point, which is this type of epigenetic inheritance in the offspring. There is a fundamental feature which we call imprecision feature, because there is an incomplete penetrance in the offspring, even if a group of offspring, they show phenotypical outcome. But we notice that the severity of the phenotype ranges dramatically. It's not like this DNA deletion induced phenotype. So here is our explanation for this phenomenon. So perhaps this concerns the fundamental difference between the DNA versus using RNA as an information carrier. Because for the DNA sequence, we only have a very limited combination. For example, we have a mutant allele or an intact allele, or perhaps a half mutant. Still, it has only limited combinations, so we can consider it as a digital signal. But for the RNA-mediated phenotype, the RNA, even for one RNA, their range of expression can range from 0 to tens of thousands. So this is more like an analog signal. So what is the phenotypical outcome of a digital signal and an analog signal? So let's take out the TV. So for the digital TV, you either get a perfect picture or no picture. But for the analog signal, the quality of the picture will depend on the strength of the signal input. So basically, this is our interpretation on this relatively unstable or perhaps incomplete penetrance of phenotype in this epigenetic inheritance. So this is just our understanding on this phenomenon. So here is the take-home message. I hope I have convinced you that the sperm RNAs, especially the sperm TSRNAs and rsRNAs and their associated modifications, can really induce epigenetic inheritance of certain acquired traits from the father. And also, I hope to say that DMT2, this molecule, is super important because it can shape the coding signatures of sperm RNAs, and it can decide whether the phenotype can be transferred. And also, I hope to say this, the importance of DMT2 might be evolutionarily conserved, because we already understand that in some of the insects, including the red beetle and fly, the deletion of DMT2 can similarly block the transmission of this phenotype. And finally, I hope to say, with this technological development, including Pandora-seq and this mass spectrometry-based modification examination, we are just at the beginning to understand the nature of sperm RNA code. So finally, I hope to thank my small group at UC Riverside and also the fantastic collaborators across continents who make this work possible, and also the funding agencies that continuously support our work. And finally, I hope to say, I hope this modern version of Lamarck's giraffe will live long and prosper. Thank you very much.